Hey guys, today's topic is topic number 3, and this is all about mitosis and DNA. The first learning targets we're going to address are talking about how mitosis maintains the constant number of chromosomes in cells. This is an important process here. And then describe the relationship that exists between the cell cycle and cancer. So we're going to talk about that one too, what the cell cycle is and how cancer is a result of cell cycle out of control. First thing we need to start talking about is the chromosomes that exist and how DNA is packaged and stored inside the cell. We know that it's in the nucleus as far as the organelle, what contains it. What we don't know is really how that information in the nucleus is then passed into different cells along the way. So to backtrack a little bit, humans have a total of 46 individual chromosomes. The key takeaway is that there are 23 from the mother and 23 from the father. We have a total of 23 pairs or 46 chromosomes. So you'll hear it referred to as one of these two ways, right? 46 total individual chromosomes or 23 pairs. DNA is copied before it's wrapped in a chromosome, so DNA is kind of loose and hanging out, this long stringy substance. And then when it's getting ready to be packaged and dragged into a new cell, the DNA is copied and wrapped into these dense chromosomes. Before the cell divides, DNA supercoils. I'm going to show you what that looks like in just a second. One side of the chromosome is the original, the other side of the chromosome is the copy, but they should be identical. So these pictures show pretty well. We have an original strand of DNA here, and then the copied strand here. The chromosomes then wrap up on themselves and then pair up in the middle with a centromere. All right, so you'll see what that is on the next slide. DNA packaging. DNA is very long, skinny, not easily visible molecule, and it's just kind of stored in the nucleus, kind of free floating around. When the cell is getting ready to divide and go into the cell cycle, which we'll talk a little bit more about, DNA is going to supercoil and wrap itself around these proteins, and that's called chromatin. When the chromatin's wrapped up on itself, it turns into this X-shaped substance known as a chromosome. So in this video, you can see what's going on here is this chromatin is wrapped up around itself. When we look inside, we can see that these little purple balls are proteins, and those are like histones, and those proteins provide some kind of structure for the DNA to wrap itself around. Some key vocabulary, because chromosomes, when we talk about chromosomes, it gets a little confusing. We'll talk about a chromosome, a centromere, chromatid, sister chromatids. So there's a lot of vocabulary that kind of refers to the same thing. And so I want to kind of make that pretty clear here. What you can see is that a centromere, that's probably the easiest one to remember. A centromere is in the center. This is a structure that basically holds the two identical things together. Okay. The next thing is a chromatid, which is an individual molecule of DNA. Remember, the chromatid is basically just half of a chromosome. If we can look at chromatids, and you can see in the video that's playing over on the other side, basically here's one on this side, and then we can do another one, different color over here on this side. So these are individual chromatids. They should be copies of each other. If they're copies, like in the video it's showing you here, when this pops up, it says sister chromatids. If they're identical copies on the same chromosome, then we refer to them as sister chromatids. If we're referring to this half here and then another half on a chromosome over here somewhere, we would just say they're chromatids. They're different, or non-sister chromatids. The chromosome itself is the whole structure. So this big thing here, that's your chromosome. So you see that kind of flying out of our nucleus here. Sister chromatids, centromere in green, and the whole structure is the chromosome. Okay, we're going to talk about some reasons why cells divide before we get into this process. Why does all this stuff need to happen? The primary and number one thing that we need to talk about is surface area to volume ratio. Really important is that as a cell grows, the amount of surface area becomes too small to allow materials to enter and leave the cell. Smaller cells are more efficient at exchanging materials, which means diffusion is really only fast and efficient over short distances. As a cell gets larger, the amount of surface area isn't large enough to take in the amount of materials the cell needs. So you can see this quick, easy picture here. We're just looking at cubes right now to kind of model a cell. But the idea that a 1 millimeter side on a cube gives us a volume of 1 cubic millimeter, 1 millimeter cubed. And we can see that our volume is 1 millimeter cubed and our surface area is 6 millimeters cubed. As it gets bigger, we can see that the volume is going to increase kind of at a faster rate than the surface area. So in this case, our volume increased how many times? We go here, well, we can say that's times 8. All right. Well, what is this doing? All right. The surface area. Is the surface area increasing at least 8 times? No, it's not. Our surface area is really only increasing times 4. To make this leap, the surface area is not increasing at a fast enough rate as the volume does, and that makes it really difficult for the cell to continue to fuel and take in the necessary components through diffusion or osmosis. Some other reasons cells divide, we'll kind of go through these quickly. Growth is an important one. Living things grow by creating more cells from pre-existing cells. That's cell theory, and that's really important that cells are able to do that. Embryonic development, creating cells in order for an embryo to grow. Embryos grow at an extremely fast rate. Fertilized egg creates cells very quickly to develop new tissues and structures. Speaking of tissues, tissue repair is important. New cells need to be created because old cells are damaged. You can think about a bad sunburn or cuts or anything like that, and these cells will replace and repair themselves once they're damaged or killed. Then asexual reproduction, creating new identical cells. We talk about our bacteria and binary fission again. We'll get to that later. That's a good example. The cell cycle is just like really a way for us to identify what a cell is doing at any given point. Cells are not always constantly dividing, so there are times in the cells, kind of you can think of as a life cycle, when they're doing other things. So we're going to break them down into 4 major phases, really. You have G1, S, and G2. G1, cell growth, is a production of new organelles. The S phase is DNA replication, and this is probably one of the ones we focus on the most. DNA replication. When does this actually happen, the doubling of DNA, getting ready to split? G2 is finishing, kind of preparing for dividing, finishes growing, and then the DNA condenses in this phase and begins to supercoil in that process we talked about earlier. So you can kind of see the cell cycle over here. It's a cycle, so you could think about it as starting anywhere, but we'll try to start it at G1. It makes the most sense when we think about it. It goes through G1, S, and G2, and it moves all the way around this way. Then we get to this process of mitosis, which has unique stages in itself that we could even further divide these up into, and you'll see what that looks like. But basically, this whole thing here, my orange arrow, all of this is all interphase. That's the first part that we'll be talking about. The next thing we really need to address is how the cell cycle can relate to cancer. Cancer is uncontrolled cell division. This results in tumors forming throughout the body. The tumor is basically a bunch of cells in one area that keep dividing. These cells can also move on through other parts of the body, causing tumors to develop in other regions. So cancer starts off in one area, and then it obviously has the ability to spread. Cancer kills by causing your organs to malfunction, and your cells just not able to stop this rapid growth. So the cells that are doing a process that are meant to provide us with all those services can't be stopped and ultimately cause serious issues. We have 2 animations here. The first one is, this top one is breast cancer cells that are actually dividing, and then the bottom one is an animation to show you what cancer cells look like, and it shows you the different stages of cell division all along the way with this massive and fast rapid splitting, which we'll talk about those individual stages. But you can see these chromosomes here. Chromosomes are these white things that start to form and get pulled apart, and all these cells are splitting at such a rapid pace.